Welcome to A&P II. Hope you're glad to be here. I'm certainly glad to be here. So you're all juniors and seniors now, and so you know the drill about the syllabus. The syllabus has a lot of useful information. Just read it and save it for future reference. We'll go over some of the high points of it, the most important things, but I'm not going to force you to review the syllabus now. You know the drill. So the syllabus is important and it has useful information. Keep it and refer to it. You'll notice that we're recording, I'm recording the class like I did in A&P I last semester. This is for two reasons. One is in the future, like maybe even this semester, these videos can be used in a so-called flipped classroom, where you watch a lecture or part of a lecture as the homework for a class, and then come to class having heard the lecture, or at least part of a lecture, and then you're ready to do some problems. So I'm not going to flip so last year I have videos, and they're up on YouTube, and I'll give you the links as needed from last year's class. And so I might say, okay, the homework for Thursday, some Thursday, is to watch minutes 10 through 20 of this video. I'm not going to teach that. You're going to see the lecture online, and then we're going to work with it. So I'm going to do that sum. You know, I'm a scientist, so I like to do an experiment, and so you are my experimental animals. I'm going to do an experiment in the class. But we're also recording this year's class for the future and for your future. If you miss a class and you want to know what happened in class, it'll be up on Moodle. If you want to review a video because you phased out there for a minute, it'll be up on Moodle. So for example, let me just show you what this might look like. So here's the YouTube channel from last year. So all of the videos will be loaded up. By the way, our videographer is Brooke, a psychology major, so she's going to be shooting the videos. So for example, if I were to do a flipped classroom for the next class, I might say, go to 2018-2, the second class from 2018, last year's class, and watch minutes 10 through 20. And then you'll be ready for the problem that we would go through in class. And you folks are probably even better at YouTube than I am, but click on the video and play it. And I'm not going to turn the sound on, but you, of course, would listen to it. And so let's just say I say watch minutes 10 through 20. You could start at minute 10. And you could speed it up if you wanted to. You could play this, I'll start to sound like a munchkin if you play it like that. But, it can go, we can speed it up. Anyway, I'm gonna experiment with this approach in this class. And if it fails, I won't do it again, that's all. So A&P II focuses on the heart and cardiovascular system, the lungs and the respiratory system, the kidneys and the renal or urinary system, and all the organs of the gastrointestinal tract and digestive system. That's the focus. A&P I, some of you already know, focuses on the nervous system and the endocrine system and the brain and nerves and muscles. But that's not a requirement for this class. A&P I is not a prerequisite for this class. So some of it, for those of you who took A&P I, by necessity will be doing the review from Self-Phys, because that Self-Phys is the prerequisite for this class. I'm sure, I know that all of you took Self-Phys, so I can rely on your remembering everything from Self-Phys. So what are we doing here? We're going to understand the structure, anatomy, and function, physiology of these organ systems. We're going to make connections between the systems. You may or may not know that the heart and the kidneys work together very closely to control blood pressure. We're going to figure that out. The heart and the kidneys work together to control blood pressure. The heart and the lungs, the kidneys and the lungs work together very closely to control blood pH, the acidity of the blood. We're going to figure that out. The heart and the brain work together, but brain is not part of this course, so we're not going to be able to go into much detail about that. We're going to focus on heart and cardiovascular, lungs, respiratory, kidney, and digestive, and make connections between those systems to try to make it a somewhat integrated whole. In another universe where A&P I and A&P II were both required and in a sequence, I would bring in more information from A&P I, but that's not the world we're living in. I'm going to ask you to make connections from cell physiology to anatomy and physiology. Anatomy and physiology will not do too much molecular, but you should remember from cell physiology, every cellular structure, every protein we talk about has a structure, has a molecular structure, a molecular mechanism. I'm not going to go into molecular physiology too much. It will mostly be cellular systems and integrated. That's the level we're at. But I might, draw in some cell physiology now and again. I probably will on a regular basis. So, and especially today, where we're building the foundation. You're going to learn anatomy and physiology is one of those courses all across the world, in the United States and around the world, where it's just well known as one of those courses with a lot of information. And that's the way I teach it as well. So you will continue your process of learning how to integrate and assimilate a lot of information and put it together. You'll work together in teams in the lab and maybe sometimes in here for, think pair shares and flipped classroom. And a lot of this is in the syllabus. 2 required texts, same text as in A&P I. So if you take both courses, the books are the same. You don't have to rebuy them. So this is the physiology textbook. It's required. There'll be reading assignments, which are in your schedule that I handed out. The reading assignments are required, and they're in here. Some of them are long because it's kind of review, and some of them are focused and short. The anatomy coloring book is the anatomy textbook. And it's not this is not a children's textbook. I mean, medical students use this book to learn and relearn anatomy. And so you don't have to color it, but you do have to learn the information from the coloring assignments. I actually recommend coloring it because that's a form of active learning and can be kind of fun. But you don't have to color it. Both of these books are on reserve at the CST desk. But and the coloring book is actually a used version that has some coloring in it. But I ask you not to color in the reserve book. Don't. If you want to color, make a photocopy of that page and color it yourself. Okay, so these are the required books, and there are required readings and coloring assignments from the 2 books. In terms of, so what are we going to be doing? You're going to be reading to learn. You're going to be coloring to learn, listening to me actively, taking notes, active listening, thinking, trying to put things together, trying to figure out what did he mean by that, and then dissecting in the lab. Those are all those are the forms of active learning that I promote in this course. You'll be asked, I want you to ask questions. I know that there's an energy barrier to asking questions, but I ask you to try to overcome it. Because if you have a question, it's almost guaranteed that several other people in the room have the same question but are just not quite there with asking it. So you're doing everybody, including me, a favor by asking the question. I don't, I make mistakes. I sometimes say things that are unclear, or maybe even, in conflict with something I said the day before, the class before. So make sure that you ask questions. And then I like to do the clicker thing, so you picked up your clickers. And I think what I'll do is, you can see the standard arrangement will be to pick up the handouts as you walk in, including the clickers, the ABCD clickers, and then just leave the clickers here. I'll bring them as needed. You don't have to save the ABCD clicker. Just leave it here at the end of class. So the assessments, where is your grade going to come from? It's going to come from exams, of course, quizzes, problem sets, and working in the lab. Those are the sources of assessment. Exams will be in this room or in the lab, closed book. Quizzes will be self-scheduled. When a quiz is available, when there is a quiz due, I will make the quiz available at the CST desk. You'll pick it up, sign it out, take it right then and there. Don't take it home, take it in the CST lobby or atrium or whatever, or a room in the CST, and then turn it in. Quizzes are closed book. So I'm trusting you to leave your stuff somewhere else. I know the temptations. You've got the answer. The answer is sitting right in your pocket. You know that answer is in your pocket. There's a real temptation to look it up. You must resist that temptation, because that's cheating. And that's lying to me. That's telling me you knew something when you didn't know it. You just knew how to look it up on your phone. So don't do it. If you see someone else with a phone out during an exam or a quiz, let me know. I'll make it. I'll approach the student. You don't have to deal with that yourself. But those assessments are closed book. The exams are in here. The quizzes are, you use time. I'll make them available, and then they're due. Typically, they're available on either on a Tuesday afternoon or a Thursday afternoon, and then they're due on a Sunday night before the CST desk closes. So that's, when a quiz is coming, I'll reiterate all this. Any questions about these assessments? So I've talked about doing some in-class, team things, flipping. Those will be experimental, and they won't count. I think they could be informative and useful for learning, but unlike the TBLs in cell physiology, which actually were part of the grade, that anything that we do as a teamwork in this room will not count as the grade. Any questions? Okay, well, if something comes up, don't hesitate to send me an email. I like to be very responsive. Sometimes you'll get a response in 5 minutes. Sometimes it'll take an hour, and sometimes it'll take a day. But I try to be as responsive as fast as I can. Because I know when you're sending a question, it's in your head. And if I can respond while it's still in your head, it's better. I try. Can't always do it. So there we are. Problem sets are due on Sunday. No, problem sets, we haven't talked about problem sets. Problem sets I'll hand out here, and they, like the quizzes, are also due on Sunday. You hand them in at the CST desk. The problem sets are open book, open note, open internet, open friends. You can ask me a question. I won't give you the answer, but I can clarify the question. So that's an open book assignment. The quizzes are closed book. The problem sets are open book. Quizzes available at the CST desk. I've already said that. Due on Sundays. Exams are in class in here. So this is all repeat. Quizzes and exams are closed book. So about half of you have already taken A&P I, so you know the drill and lab, and half of you have not. I'll introduce you to lab this afternoon. So there is a lab meeting at 1 o'clock today where we will partition the class. This is too big a lab. This is too many students for one lab section. So we're going to partition into 2 lab sections, one in the afternoon from 1 to 3:30 or so, and one in the evening on Thursdays from 7 to 9:30 p.m. We'll run a lottery to help partition and then make adjustments, and the adjustments have always worked. I sent you an email, I think, about this about a week ago. So anatomy lab, I mean, this for many Earlham students, this is the sort of the peak experience to work on the body. You know, for some Earlham students, it's like, okay, I don't know if I can do this, but for many Earlham students, it's, you know, it's just an amazing opportunity. It's pretty unusual for a school this size to have this kind of anatomy lab, which much more typical is pictures or digital only. We use digital too, but not only. Or animal dissections, which are a pale learning experience compared to dissecting a real human being, at least as far as human anatomy is concerned. If you're interested in dogs, doing a dog dissection is pretty informative. We do human dissection. It's an amazing opportunity. You are becoming part of a community of people who have worked on a body, a fairly elite community of students and nurses and physicians and physical therapists who have worked on a human body to understand anatomy and physiology. I will guide the dissections, but I will not be doing the dissections. You will be doing the dissections. I will be guiding. And that's also unusual. Many liberal arts colleges that do have an anatomy lab are structured where the professor and the TAs do the dissection and get it all set up so it's real pretty, you know, very informative, nicely done, because they've done it before. And then they demo it to the students. Now that's not the way we do it here. You're gonna do the dissections. They're not gonna be perfect, but they're gonna be more engaged, more informative for you. Okay, so here are the systems we're going to talk about. I'm going to transition into actual content, like now. Yeah, like right now. So this was a very quick overview of the course. Any questions, please? I'm sure I missed something from the syllabus, but the syllabus is pretty thought out, so the information is there. I do try to engage in an interactive lecture. I do most of the talking, but not all of the talking. So you'll see clickers and think fair shares and 2-minute write things to get a conversation in this class. It's not so easy to have a conversation with 30 people, but I'm going to do some of that. Okay, so we're starting. And some of this is going to feel a little bit like, drinking from a fire hose, right? So much information coming at you so fast that you can't keep up. But you can keep up. Sometimes it means catching up by doing homework. And sometimes it means just being fully present here now to stay with me. I try not to talk fast, and I fail. So I'll try to slow down as needed, but I do talk fast and I go through material fast. It's your responsibility to slow me down as necessary for you. There was something else I wanted to say about class. Oh, homework. So we've talked about assessments and, so taking a quiz is you do it at home. I mean, not if you do it downstairs here. That's outside of this classroom. But problem sets and quizzes are not the only homework. This is a 4-credit class, and one of those credits comes from the 2.5 hours or so of work that you'll do in the lab. And this lab, unlike Self-Phys, does not have a poster session, it doesn't have a lab report. It's really just being fully engaged in the lab process. So it's pretty easy. Show up on time, participate, you get the full credit. The 3 credits of the course come from learning in this room and learning at home. And so I think, the rule of thumb for a 3-credit course, because this is a 3-credit course plus the 1-credit lab, the rule of thumb for a 3-credit course is that for the 3 credits, you're spending 3 or so hours in this room a week, and you're spending 6 to 9 hours per week doing homework. So that means reading, thinking, retaking your notes, reviewing the video if necessary, rewriting your notes, reviewing the video. So a lot of your learning, 2/3, 3/4 of your learning, is not in this room, it's doing the work that's required. And it's not just the problem sets and the homework, the quizzes, getting ready for quizzes, it's keeping up with every single class. That's the way I'm going to assume. And if it means I'm assuming you're going to do the reading and learning from the homeworks. Okay? So, let's start. We're going to start fast. We're going to do a very, very fast review of some of the key concepts from Self-Phys that we are going to be using over and over and over again in this course. And some of those key points are related to transport of solutes, which you may remember from Self-Phys, and some of those are related to signaling mechanisms, which we will do on Tuesday. Okay? So this is all review, which means I'm allowed to go fast, but it might mean that you have to go back to your Self-Phys notes or your Self-Phys book to fill in the gaps. Okay. So we're talking about transport of solute, ions or small molecules, through membranes. And we have 4 main ways that this happens. The solute might simply diffuse through the membrane because it's amphipathic or hydrophobic enough that it can just go through the hydrophobic part of the membrane. Sometimes things go through a channel, and usually in this case we're talking about ion channels. And we're going to be talking about some very important ones in the heart, in the kidney, in the respiratory system, all over the place. We're going to see ion channels a lot. We're going to talk about passive transporters that are similar to channels in some ways and different from channels in other ways. We're going to get to that in a minute. And we're going to build the gradients that the cells use using active transport. What's the form of energy? What is the energy that is going to be most common in this course? ATP. So, like I said, some solutes can diffuse right through a membrane. And for this course, the ones that are important are oxygen and carbon dioxide. We're going to get to that in the respiratory system. They just go through the membranes. They go wherever they want. They diffuse through membranes, oxygen and CO2. All the other important solutes that we talk about, sodium, potassium, chloride, calcium, bicarbonate, amino acids, they cannot just diffuse through a membrane. Membranes are impermeable to those solutes. So we need transport mechanisms, pumps, channels, and passive transporters to move them where we need them to go, we being the cell, or the organ. So let's talk for a minute about passive transport. And you know that the active transports have built up gradients. We'll come to that in a minute. But you know from self-is that active transporters have built up gradients, and now we're going to talk about passive transporters. Let's start with ion channels. Hopefully the word voltage-gated sodium channels and voltage-gated calcium channels and ligand-gated non-selective cation channels. Hopefully those words are triggering some memories from self-is or from A&P I. Ion channels are transmembrane proteins that undergo conformational changes between a closed state and an open state. And when they are open, ions go downhill, go down their concentration gradient that was set up by some active transport process. So this is showing ions moving from, now it's always important with membrane cartoons to figure out, okay, which is the inside and which is the outside. This is the outside of the cell, and this is the inside of the cell. And I have to admit, this is reverse from what is typical. This is a figure from your textbook. This, almost always, I will show the inside down and the outside up. But you have to get oriented. Right? So in this case, we're talking about ions moving out. So that it's always important to know which direction are things moving. So in this case, the concentration of whatever is moving, whatever this arrow represents, the concentration was higher on the inside than on the outside, so that when the channel opened, the ion moved out. What's the most likely candidate for an ion that is higher concentration on the inside, lower concentration on the outside, so that when the channel that is permeable to that ion opens, it flows downhill from in to out? There's one ion that is in my mind. A proton what? Well, a proton, so an ATPase is not a channel. So right there, I know you're off track, because an ATPase is a pump, and we're talking about channels. But this is good because, so when we talk about ATPases, we're talking about primary active transport that set up the gradient. In this case, I'm talking about a channel that is using the gradient that was set up by an ATPase. Protons, now, protons, that's not the ion I was thinking of, but that is a correct answer. If I asked you this question on an exam, I'd have to give you credit, because the pH on the inside is typically lower than the pH on the outside. Now I'm doing pH, right? So pH on the inside is lower, that means the proton concentration is higher on the inside. So proton is a correct answer, but it's not the one I'm thinking of. So, Potassium. Potassium is the ion that has a high concentration on the inside and a low concentration on the outside. But this was an example of the kind of conversation that is very useful. A partially correct answer that allowed me to elaborate a little bit. So thank you for giving me that. What was a pH or protons was not exactly what I was thinking of, but it was a correct answer. ATP ACE was the wrong part, because that's not a channel, and that was a good thing for us to be talking about. Okay, the other type of passive transporters are called passive transporters. They're not channels. They're uniporters, I think is the word that I use in self-viz. They catalyze facilitated diffusion. That's what they do. So in this case, so let's see, let's get oriented. This is from your textbook, so the intracellular is up here and the extracellular is down here. We have a solute that binds to the transporter. What is this arrow? What is that arrow signifying? This arrow right here. Conformational change. Binding, well, diffusion and binding. Conformational change. Unbinding or dissociation and diffusion. And so in this process, the solute moved from out to in. And one of them that we'll be talking about is a glucose transporter. And we're going to see this in the digestive system. It's actually everywhere. All of your cells have it. It's very important in your brain and your muscle too, but that's A&P I. But this is A&P II. We're going to talk about how you get glucose from your food into your intestines. And this is one of the important mechanisms. Okay, we're going to do a little question about the difference between a channel and a transporter in just a minute. What I wanted to do is just remind you, okay? So, like I said, we're not going to be doing too much molecular physiology or molecular mechanisms. We're not going to be doing too much of that. And so you're going to be seeing lots of these blobograms, right? The protein as a blob. But you know that proteins are not blobs. Proteins are proteins with primary, secondary, tertiary, and quaternary structure. So I'm not going to show you molecular models of these proteins very often. I'm going to rely on your knowledge from cell physiology to, in your mind, whenever I show something like this, you're reminding yourself, oh, what Bob sees in his mind is something like this. Right? So this right here is a side view, a side view of this protein, the glucose transporter. So this is the blobogram, which you're going to be seeing a lot of. And this is this is a cartoon also. It's a cartoon of it's a ribbon cartoon of the protein. This has a lot more information than this. We're not going to be talking very much about the protein, about the information embedded in this cartoon, but it's there. It's always there. And sometimes I'll bring it out explicitly. So this is a side view. So the membrane is this way. What's this second? Let's do a little review. What's this name of the secondary structure? Alpha helix. This is almost completely alpha helical, this protein. So this is the side view, and if we rotate it, I think if we rotate it this way, this is a top view of the protein. And now you can sort of see some of the, anybody know? I'm not going to show off. So that's the side chain of an amino acid that happens to be tyrosine. Okay, I will show off. That's tyrosine. And in here you can see a pocket where, what do you suppose happens in this pocket of this protein? Glucose binds. And so if we could go through the molecular mechanisms or even the atomic mechanisms of how glucose binds to this protein and then undergoes the conformational change to do this. But we're not going to get into it too much. Some of you will be disappointed that we're not going to get into molecular mechanisms. Most of you are probably pretty relieved. It's there though, all the time. So here is the glucose transporters, side view, top view. Here is a voltage-gated calcium channel. So the blobogram of a channel and the ribbon diagram of a very important channel that we're going to see over and over again, a voltage-gated calcium channel from the side and from the top. What do you suppose that green sphere is supposed to represent? That is the binding, well, the green sphere is calcium. That's the calcium ion sitting in the channel of the voltage-gated calcium channel. This is side view, top view, so we're looking down the hole, and so that green thing is the ion in the channel, right? And in the blobogram, we don't even see the green thing in the channel because all we got is the arrow. Hopefully these are ringing bells. These concepts will be used over and over again, and I'll be assuming that you know what I'm talking about. So let's do what I call a 2-minute write, and you can work with a neighbor if you like. Let's spend 2 minutes. What are similarities and differences between ion channels and these uniporters, the facilitated diffusion transporters? And not that, so remember, so for example, if you say, well, voltage-gated calcium channels carry calcium and uniporters carry glucose, that's not strictly a true answer because uniporters carry a lot more than just glucose. So don't get too specific. Think about the general truths of the similarities and differences between these two classes of carriers, channels and uniport transporters. Let's take a couple minutes, and then we'll talk. Either a difference or a similarity. I'll run back and forth. You don't have to, just because I'll stand here in the middle, tell me which way to go, and are you talking about a similarity or a difference?
I have a difference.
Okay.
Ion channels allow for a high volume of ions to move, whereas facilitated diffusion carries a few at a time.
Excellent. Okay. So let me say it a little bit differently. Ion channels have a very fast rate of transport. And transporters, or let's call them facilitated diffusion, much slower. The idea here is, so let me go back to the blobogram. A channel, when the channel is open, ions flow, and 1,000 ions could flow for every opening of the channel. The channel opens, 1,000 ions flow, and then it closes again. And then it might open again, and 1,000 more, or 10,000 more flow, and then the channel closes. The transporter, 1 or 2 solutes move for every conformational change. So in this cartoon, it's 1 solute is moving for each conformational change. And the bottom line here is for you to remember, or maybe now learn, protein conformational changes are slow compared to diffusion. Diffusion is relatively fast. Protein conformation changes are relatively slow. So when this protein undergoes its conformational change and the ions diffuse through the channel, thousands can move per conformational change. When this protein, only 1 ion is moving per conformational change, so it's real slow. Grant, did you have another answer?
Yeah, for similarities.
Okay.
They both are specific for solutes.
Good. They both have specificity. A voltage-gated calcium channel is very specific for calcium. The glucose uniporter is somewhat specific for glucose and sugars, but it doesn't transport, sodium, right? It transports sugars. Some things are absolutely specific, like the voltage-gated calcium channel or voltage-gated potassium channel, and some are relatively specific, like the glucose transporter is specific for glucose, but it also transports xylose, and it can transport fructose, so it's got somewhat lower specificity, but that's an important similarity. Okay, good. Another one, either kind. Lots more of both. Some of them are so obvious that you're going to say, oh, why didn't I just say that? Isaiah.
Binding versus free flowing.
Okay, so is that
A difference, I guess.
So you're saying in one case there's binding and in the other case there isn't?
Well, I'm just saying, when the ion channel's open, they're flowing down a concentration gradient, and then
They are flowing, but they both have binding. So this is a flaw in the channel blogogram. When ions move through channels, they are binding to sites along the way. They're not just flowing like this blogogram cartoon would suggest. So this blogogram cartoon gives you the impression of just diffusion, but it's diffusion and binding in both cases. The difference is that binding here is, well, let's just say over here. So the channels, the binding has a very fast off rate. It unbinds very fast, so that it binds, but then it unbinds, and so it can get through fast. The transporter has a very slow off rate, or much slower off rate, so that it stays bound longer. Is that clear? The transporter, binding of the solute is longer, but they both have binding. That's a key knowledge point that is obscured by this hyper simple cartoon. Grant?
I just had a question based
Yeah.
on that. So for the channel then, since there are binding sites, does that mean that it's kind of like single file, they follow through just quickly?
They do flow single file. In fact, there's a whole field of study of the single filing of ion channels. In fact, they're both single file. This is single file of one channel, one ion at a time. And this is single file because it's single. Andrew?
They move down the gradient and they don't utilize ATP.
Okay, yeah, they're both passive. They're both downhill. Does everyone know what I mean by downhill? It's going down the concentration gradient. No energy is necessary for this process. The energy was spent by something else to establish the gradient.
This is a question more than anything.
Okay.
I know that channels are turned on and off, but can ion transporters, passive transporters, be turned on and off? Is there like an inhibitor?
There is, but this is a good point. So ion channels are typically activated by something with voltage or a ligand. Transporters are much more likely to be constitutive. They're just running. They can be turned on, the activity can be accelerated by phosphorylation, it can be accelerated by drugs or inhibited by drugs, but the transporters are typically just going. But they can be modulated. Channels have a stimulus that opens and closes them. That's a real good point. So that's a difference. So let's just say that facilitated diffusion transporters are generally constitutive activity. Typically. Kendra?
So, for example, for the facilitated diffusion transported rate, it only conformationally changes when something binds to it.
That's right.
Okay.
Well, that's kind of right. So when something binds, it undergoes the conformational change that allows the solute to go out. But then, even in the absence of the solute, it can undergo the conformational change back to the original position. So in some of the conformational changes depend on the binding, but some of them don't. That's a trick question. Okay, other similarities or differences? Some real obvious similarities. What are they both? Somebody besides Grant. Matt.
Passive transport.
Okay, well that comes from the name.
They both undergo conformation.
That's right there on this,
Yeah.
that is super obvious, yeah, that's right there on the slide, but I'm thinking of something else. I've said them, you know, maybe the super obvious ones are too super obvious. Tyler?
They're both transmembrane proteins.
They're both transmembrane proteins. That's just super true. Anything else? Anybody else want to add to this? So let's see what I came up with when I was trying to answer the questions. So, ion carriers, passive, high to low concentration, 1 or 2 ions or solutes per conformational change. I said that, didn't write it down. This is getting at the idea that these are slow compared to these. These are fast compared to these. Okay, so let's just do similarities. They're both passive. They both have sites. We didn't say. They both undergo conformational changes. That's a similarity. Slow versus fast. Or some of the other differences. Slow versus fast. Constitutive versus stimulated. That was I didn't put that on, but that's true. Okay, so this was a useful demonstration of the kind of conversation that these kinds of questions promote. The Moodle, so the PowerPoint will be available on Moodle at the end of every class. If I forget, which will happen at least once every semester, so feel free to say, Bob, where's the Moodle? I mean, where's the PowerPoint? And I'll apologize and I'll put it up as soon as I can. So now we're going to talk about the pumps, the active transporters that establish the gradients that the passive transporters, channels, and uniporters use. Let's take a break. Let's take a few-minute break. I'll do that for every class. We'll take a break for, I don't know, 4 or 5 minutes. I'm going to send an email to facilities and tell them that this room is too hot. And then we'll get to the active transporters. ATPases and that are primary active transporters and co-transporters that are secondary active transporters. Active transporters, using energy to establish a gradient. The most important one is the sodium-potassium ATPase. This couldn't be blobbier than this, right? This is this is the blob animal, right? A membrane protein that hydrolyzes ATP, pumps sodium out, and pumps potassium in, and makes it and establishes that gradient that I asked you a question about before, with the high concentration of potassium on the inside relative to the outside, and a high concentration of sodium on the outside relative to the inside. Primary active transport, why do I call it primary? What is the thing about this that makes it primary? It hydrolyzes ATP. That's the definition of primary active transport. So here's the life cycle of the sodium potassium ATPase. Let's tell the story. Here are the pictures. This is the cartoon pictures. Let's tell the story together. So let's start here. So this is the inside, this is the outside. Here's the membrane, here's the blobogram. ATP has bound. So remember there was ATP on the inside. Where did that ATP come from, self-phys students? Organelle, I'm thinking of an organelle. Mitochondria, oxidative phosphorylation. We're not going to talk about mitochondria and oxidative phosphorylation too much. It's there, it's always happening. Your cells have ATP. If they don't, they're dead. So ATP bound. Sodium ions, 3 of them actually, bind to a site inside the protein. In your mind, you should be thinking, oh, there's probably alpha helices and there's specific residues, maybe tyrosines, who knows what. I'm not going to get into it, but it's all there in the back of your mind. Okay, so what does this arrow represent? Diffusion and binding. What does this arrow represent? Yes, there is a conformational change, right? The inside sort of became somewhat open here. Something happened to this inside part of the protein. What else happened? There's 2 things. Conformational change and hydrolysis of ATP. Right, so you had to notice that, oh yeah, ATP got hydrolyzed. So that was what this enzyme did. Okay, what does this arrow represent? More a major conformational change from sort of an inward facing, right, with the big mouth facing the inside to the big mouth facing the outside. Major conformational change. But no hydrolysis of ATP, because that happened here. What does this arrow represent? There's 2 things that this arrow represents. Unbinding and diffusion. Good. So sodium has just moved from inside to outside. And ATP was hydrolyzed. Okay, so there's an arrow that goes from here to here. So what does the arrow, well, what does this arrow represent? It's right here. Diffusion and binding of potassium. So what happened? So this conformational change made the sodium ions less likely to be bound. Something happened to those binding sites that lowered the affinity of those sites for sodium. So they didn't want to stay bound, so they unbound, right? And that's cartooned here by these binding sites that are, sort of nice spots for spheres to bind, now become too small. That's what this cartoon is illustrating. They're too small, binding is crummy, so they unbind and then they diffuse. It also turns these crummy other binding sites, the sites for potassium, into nice, happy binding sites for potassium. And so potassium diffuses and binds. This conformational change sort of semi-opened the pump, and then there was another following conformational change that lowered the affinity of sodium, sodium unbound, increased the affinity for potassium, so potassium binds. Kendra?
Is that before or after the phos?
Okay, so the potassium, I'm trying to remember my molecular physiology, the potassium binds before the phosphate dissociates. So this step happens, then the phosphate dissociates from the pump, triggering What's this? A conformational change that lowers the affinity for potassium. Potassium unbinds, ATP binds, and that reconstructs the original binding sites. We'll go back to this one. I'm pretty sure I walked through this story in Cell Phys. I'm not going to ask you for the molecular details now because you learned it for Cell Phys, but it's there in the background. And knowing that series of bindings, hydrolysis, conformational changes are what generate the function of this protein to pump sodium out and to pump potassium in. This protein is going all the time. It's constantly maintaining those sodium and potassium gradients. But it's not the only ATPase that we're going to be talking about in this course. We're going to be talking about calcium ATPases. And there's going to be 2 of them. So here's a cell. Here's a nucleus. I could go on, but I'm not going to. Cell physiology. There are so again, the blobbiest blobogram. There are calcium ATPases. What goes on the 2 sides of this arrow for an ATPase? ATP is hydrolyzed to ADP. What goes on that arrow for a calcium ATPase? Just gave you the answer. Calcium. Calcium is pumped out. This protein has a name. It's called the plasma membrane calcium ATPase. Because it's a calcium ATPase on the surface membrane. Many of the cells, all of the cells that we also are going to that we're going to be talking about, have a endoplasmic reticulum. And we're specifically talking about the smooth endoplasmic reticulum now, not the rough ER. So we're not talking about protein synthesis and ribosomes on the rough ER. We're talking about the smooth ER. There are, so what goes here? ATP. What goes here? ADP. What goes here? Calcium. Calcium is pumped out of the cell, or it's pumped into the lumen of the smooth endoplasmic reticulum. This protein also has a name. Now, you'd like to think that this is the smooth ER calcium ATPase, but in this case, the S actually stands for another word, sarcoplasmic endoplasmic reticulum calcium ATPase. But the point is, it's a smooth ER. It's a protein resident on the membrane of the smooth ER that pumps calcium into the lumen, out of the cytoplasm. So typically, where is calcium concentration high in a cell, or in an organ, in a system? Where? Okay, but that's true. There is calcium in muscle, but I'm thinking in this place where. So remember, sodium concentration is high outside the cell, and so calcium concentration is high outside, It's very high on the inside, and it's very low in the cytoplasm. Calcium concentrations are super low, nanomolar. Calcium concentrations outside the cell and inside the ER are millimolar, a million times more concentrated. And there are proton ATPases that we'll talk about later. We'll talk about those later in the course. But I wanted to introduce the sodium-potassium ATPase, because that's the most important one, and the calcium ATPases, because they're almost as important. We'll talk about proton ATPases later. So here's a blobogram of a proton ATPase. Let's just review a bit. What are the blobs? What concept is embedded in these blobs? Say again? I'm sorry, I didn't hear. Do we see primary structure? No. Do we see secondary structure? No. Do we see tertiary structure? No. What do we see though? We see quaternary structure. We see subunits of multiple proteins blobbing together to form the functional protein. So just, when I talk about the calcium ATPase, I draw it as a single blob, but it's got multiple subunits. All that protein physiology that you learned in Cell Phys is underneath you, and it's embedded in there as well. Okay, one more main concept I think for today, and then we'll be done for today, and that's another form of active transport, co-transport, or secondary active transport. And so we're going to talk about, this one is going to be important in the digestive tract. This one is important in A&P I, so we're not going to talk about it. This one is important in A&P I, so we're not going to talk about it. But there's another one that isn't even mentioned here. Well, let's talk about the sodium glucose co-transporter. I'll do that one over here. Anybody remember from Cell Phys the difference between primary active transport and secondary active transport?
Secondary uses an established concentration gradient to pump another solute across or against this concentration gradient.
Up its concentration gradient. That's exactly right. Primary uses ATP as a source of energy to establish a gradient, and secondary uses another gradient to establish a gradient. So for example, in the sodium glucose co-transporter, glucose, which is shown in purple, is being pumped into the cell, and the energy for that pumping of glucose into the cell. Why is it pumping? Because the concentration of glucose is low on the inside. Low on the outside, sorry. We're pumping glucose into the cell, low on the outside and high on the inside. So to move glucose from low concentration to high concentration, we have to pump it. That takes energy. Left to its own devices, glucose would flow down its concentration gradient from inside to outside, and that happens sometimes too, but that's not the story being illustrated here. Is the energy for this glucose pumping coming directly from ATP? No, this protein does not hydrolyze ATP. It's coming from the co-transport of sodium. Sodium is flowing down its concentration gradient from high concentration to low concentration. That is a favorable reaction. And glucose is being pumped. So the energy is coming from the sodium gradient. Where did the sodium gradient come from? The sodium potassium ATPase, from the ATP hydrolyzed by the sodium potassium ATPase. So that's why this is secondary. It's not using ATP directly, it's using ATP indirectly. It's using a gradient, sodium, to pump glucose up its concentration gradient. We're going to see this in the intestines. I'll remind you of it because it'll be a few months from now. So the idea here is that we have a high concentration of sodium on the outside, we have a low concentration of sodium on the inside. In some settings, we have a high concentration of glucose on the inside and a low concentration of glucose on the outside. By sodium going down its concentration gradient, the energy of that downhill flow of sodium is harvested by this protein to move glucose up its concentration gradient. And are there protein conformational changes in this story? For sure. Binding and unbinding? For sure. All that stuff is there in this protein. Maybe we'll start on Tuesday with a key difference between a passive uniporter and this, because this looks a little bit like uniporter, conformational changes, opening and closing on either side, but there's some key differences. Stay with me just for 5 more minutes. Let's do a clicker. I want to illustrate how this will happen well. Oh, thank you. Oh, wait. Okay, so I gave you the answers. All of the above. Right, so you show me all of the answers. Sorry, I didn't do that one. Let's do another one. Oh, wait, before we go on to this one, which of these transport mechanisms require protein conformational changes? Well, they all do, okay, that was pretty easy. And then I gave you the answer before you even had a chance to figure it out. Okay, which of the following, directly or indirectly, provides energy for making or maintaining these gradients? This one's trickier. I'm glad I didn't screw this one up. Let's go through it. Is A true? Yeah, sure. Primary active transports use ATP. Is B true? Sure. Those are the food sources that mitochondria use to make ATP. So that's indirectly, that's definitely a true statement indirectly. Is C true? Yes, it is. NADH is the source of high energy electrons that the respiratory chain uses to make the proton gradient that makes ATP. This NADH, this is asking you to remember something from self-is. NADH is the source of high energy electrons for the electron transport chain in the mitochondria. It's one of them. You also might remember, wait, what about FADH2? Yeah, that's the other. Nuclear fusion in the sun.
No.
Yeah, why? So that's a correct answer. Why?
Oh, I don't know. I just thought that it's not correlated to the so I don't know.
What do we need nuclear fusion in the sun for? Photons, which are harvested by plants to make, now we got photosynthesis going on, make sugars, which we eat, which become the fats, sugars, and proteins for our ATP, that our mitochondria use to make ATP. So we all of our energy ultimately comes from the sun. So that's just sort of one of those things to know. Okay, we'll start on Tuesday with this 2-minute lecture. So as homework, think about in what ways So this is a different question than the one I posed before, so I'm gonna ask you two questions. In what ways are primary and secondary active transports similar and dissimilar? And in what ways are passive uniporters and secondary active transporters alike and dislike? And we'll start with that on this one, Tuesday. Okay?